Okay, so chapter 3, the skeletal system. A lot of good chemistry here when we talk about the bones, right? They got to be hard because they got to protect all this soft stuff inside, and then give muscle something to attach to. But they need to be more than just hard. They need to have a little bit of flexibility, otherwise they just snap. So we need to get into the chemistry of ionic bonds for all those minerals, the calcium stuff that makes up bones, but also covalent bonds, the protein part that gives us some flexibility. And then, of course, bones are the framework for the body. It's what all the other stuff sort of hangs off of, and that needs to grow. It's primarily our bones that grow that allow us to get taller. Now, we could have easily talked about all this stuff and did talk about cell division with the skin, this constantly replacing itself through the cell cycle. But we get into that again with the bones, and this time get into the DNA itself. So in order to make two cells, you have to have DNA to go in both. So we need to get into how does DNA copy itself. And it's a surprisingly simple process. Here we go. Well, let's start our discussion here with chemistry. And the hard part about chemistry, I think in my opinion, is the scale. Is down here at the nanometer scale. So we've looked at millimeters before, and a micrometer is 1,000th of a millimeter, and a nanometer is 1,000th of that. So this is too small to even see in a microscope. So in a sense, it's kind of imaginary. Really, any chemistry usually starts with the discussion of an atom. If you can break up like an iron bar into the smallest piece of something that's still iron, that's an atom. And so circling around all atoms are these negatively charged particles called electrons. And they have a negative charge. That negative charge becomes fairly important. If they were to gain an electron, then they would have an even more negative charge. So you have an atom or even a molecule that gains an electron, then it becomes a negatively charged ion called an anion. If it were to lose one of its electrons, then it would have a less negative charge or positive charge, and then it would become a cation. All right, so ions in general are something we deal with quite a bit in biology, and they become very important. But let's just leave it there for now. When we talk about ionic bonds, we're going to have to talk about ions. All right, inside the nucleus then, we have some particles that are positively charged, and those are the protons, and they have a positive charge. Gaining and losing, this is not applicable. Unless you have like a thermonuclear reactor, you are not going to change the number of protons. Protons are kind of what give each molecule its flavor, or each atom. And then there are others with no charge, and these are big heavy particles called neutrons. And they have no charge. But they do add mass to the atom. Do you gain and lose them? Occasionally. So this is somewhat rare. And what happens if you do is you form an isotope. We'll come back to isotopes, I think, in chapter 12, but I wanted to mention it here. You can gain and lose them. It doesn't have a lot of biological significance, but it does have some chemical significance. Well, let's put some atoms together and form compounds and molecules. So I want to talk about compounds that are formed when you make ions first. So the first type of bond we'll talk about is an ionic bond, here with sodium symbolized Na and chloride symbolized Cl. This is table salt, sodium chloride. And if you remember, each of these shells, these little circles, holds electrons. The first one holds 2. The next one holds 8. And the one around that can also hold 8. We have 2, then 8, then 8. In the case of sodium, just in kind of its natural state, it should have 1 electron in that outer shell. But since atoms like to be stable with 8, that idea of the octet rule, then usually what happens is it gives that 1 electron up to chloride, because chloride has 7 of the possible 8 electrons in that outermost shell, this one out here. And so it's going to add an electron right here. So we're going to steal it from sodium and give it to chlorine. Chlorine now has an extra electron, so it's going to take on a negative charge. You might remember from up here that we call an anion. Sodium now, over here, is missing an electron, and so it has more positive ions. Remember, inside the nucleus, there are those protons that give it a positive charge, and now there's one more proton than electron, so that sodium takes on a positive charge and becomes a cation. Now, you've heard the phrase opposites attract. These two ions now are very strongly attracted to one another and form an ionic compound. And so this is true of salts and other electrolytes. Once you dissolve a salt in water, it breaks up into these individual ions that can't stay together. Also true of acids and bases, and it's true of the minerals in your bones, so not all the bones, but the mineral part of your bones. Those are made of ionic compounds having to do with calcium. Most ionic compounds dissolve easily in water, but not as much the calcium ones. Well, let's move over to molecules. And both of these here, the water here and this organic compound over here, the type of bond they are is a covalent bond. We're going to work later to talk about different types of covalent bonds, but for now, what we need to know is a covalent bond is when adjacent atoms share their electrons. And so this little double arrow is indicating that this carbon atom has an electron circling it in that outer orbital, and so does this one, and they're going to share that pair of electrons between them, and we symbolize that with just this single straight line. That means they're sharing a pair of electrons, and that's a covalent bond. You could share 2 pairs of electrons. Let's say we had 2 carbon atoms. If we shared 2 pairs, then we'd show it as a double bond with 2 lines like that. Covalent bonds, pretty simple. The magic about them is they're really stable. Unlike ionic bonds over here, they don't readily dissolve in water. And so if you put them in water, it's going to stay together. And since our body is mostly water, we want our molecules that make up most of our body to stay together. So stable, they're a little more flexible. You can move those carbons around and sort of wiggle them, and they still keep all the chemical properties together. So we'll come back to a bunch of those later, but that's the basics. You have ionic bonds and covalent bonds, and those are kind of the foundation of all the types of compounds we have in the body. Moving to the bottom of the chemistry page, I want to focus now on biomolecules. Biomolecules are organic molecules, have special functions in the body. All organic molecules contain carbon and hydrogen, so not all molecules fall into this, but a lot of the ones in the body do. There are 4 that we'll focus on, and in this particular chapter, we want to focus on nucleic acids. May not be a term you've heard before, but I think you're familiar with DNA, and the NA in DNA stands for nucleic acid. Each nucleic acid, along with all of these, have this base structure, also known as a monomer. and these are going to be put together into larger structures. And the monomers here, this one pictured is a nucleotide. And DNA has 4 nucleotides: adenine, thymine, guanine, and cytosine, and we'll mostly just use those letter abbreviations. Later when we get to RNA, we'll also add uracil. So those are the nucleotides. They all have some specific parts to them. One is a phosphate. The next, right here, is a sugar. So that sugar is deoxyribose in this case, and then it has this nitrogen base, and that nitrogen base varies, and that's what gives each of these a different chemical character. So the function of nucleic acids varies a little bit, but for DNA, our function is simply information storage. So eventually we'll take on all 4 types, and it's important that you know what each of them do, so we'll talk about functions of each. We'll add to this list, but DNA stores information in the order of A's and T's and G's and C's. So the nucleotide order or sequence stores information about how to make proteins and basically how to operate a cell. Just like all those 1s and 0s inside your computer or phone tell it how to run its software, this is our genetic code. And where we'll focus on next is how does that code copy itself in order to get into new cells. We'll make one quick stop by cell structure and function. We're not going to talk a lot about these right now, but we do want to talk about the scale of this. So this is at the micrometer scale. A micrometer is 1/1000th of a millimeter. So pretty small. Remember, a millimeter is about the thickness of a dime, so this is 1,000th of that. This cell is probably about 100 micrometers across. This kind of represents a generic human cell. So it's about 1/10 of a millimeter. Big enough where you could see it under a microscope, but not with just a magnifying glass. So that's about the size we're talking about. Still kind of imaginary. And all we want to just mention along the way here is DNA wrapped up here in the nucleus. So that's our first structure that we'll learn. So we have our DNA contained in a nucleus. It's got this double membrane around it. So there's that. And when DNA is copied and makes more DNA, that's a specific process that we know as DNA replication. So we're going to talk about how the cell does that in that tiny little space of the nucleus. We can skip forward a couple of pages to the growth and reproduction, and this is where we see a little bit of the DNA itself right here. Give us an idea of the scale of this. The chromosomes themselves, these guys here, are something that you can see in a microscope, so that they're kind of on the order of micrometers, but the individual nucleotides are nanometer scale, so it kind of is in this really small range. And in an ideal setting, this process of duplicating DNA takes about 8 hours. Important to fit this into bits from chapter 2, so we focused on the cell cycle in chapter 2, if you remember the cell cycle. And that had mitosis as kind of the star of the show, and we focused a lot on that. But now we're going to focus on this right here, the S phase. So we did some growth and then a little more growth, but we need to come back to how does the cell make more DNA? Does it just have a little copy machine in there? What does it do? So there we're going to kind of zoom out and look at the details, or I guess really zoom in. Here inside the nucleus, this one shows 2 chromosomes. We're going to ignore this one for now and just say, looking at this chromosome, if we were to really unwind it and just look at this little tiny sliver of that chromosome, then we'd see a bunch of coiled up DNA, and that splits into actually two strands that could be split apart. And those are made up of nucleotides that are all joined together, but those functional parts, the nitrogen bases, we're just symbolizing as A's and T's and G's and C's. And so we have this big enzyme. So it's a big protein that comes through and moves in that direction. And that enzyme is DNA polymerase. Which is a name that makes a lot of sense. Enzymes all end in ASE. And this enzyme, its job is to make DNA polymers. So it kind of makes sense that it would be DNA polymerase. There we go. And his job is to come in and take free nucleotides. So there's a bunch just floating around in here, a bunch of A's and T's, a whole bunch of them, and a whole bunch of C's. And they're just floating in here, and DNA polymerase grabs those and says, okay, I'm going to match this up. If I see an A over here, then I'm going to match it up with a T. Same thing here. C's match with G's, G's match with C's. And I'm just going to keep doing that all the way down the line from one end to the other. And then another DNA polymerase is going to move down this strand. They move on the old strand from the 3' end to the 5' end. So they construct a new one in the opposite direction. And what that gives us here is sister chromatids, which you should recognize is what's down here in this cell that gets split apart in the anaphase part of mitosis. But we're kind of backing up to where did those sister chromatids come from? How did they get made? This is how they get made. So these two strands, this one and this one, these two separate chromosomes are identical, but they're joined at the hip by this thing called the centromere so that they don't get separated from each other until it's the exact right time. And that's actually most of the process. There are other details we need to go into about DNA replication, but that's the basics. All right, well, let's turn to the physiology section where we take a bit of a broader look at all these systems and how they fit together. I'm going to go to the bottom of the page where we see the bones right here. Let's first talk about that mineral component. So the bones themselves have lots of minerals in them, things like calcium ions with a 2+ charge and phosphate ions, the 2- charge. And that stores a lot of those minerals that can be then transported into the bloodstream. And if the blood has extra, they can put them back into the bones. And so bones are an important ion storage. If we need more calcium, we can just pull it out of the bank that's in the bones. At the ends of the bones, at least in the long bones, there are these growth plates. And there's also a bunch of red bone marrow. And both of those places is where mitosis occurs. So if mitosis occurs here in these growth plates, then the bones get longer. And if mitosis happens in this red marrow, then that's happening all the time to crank out a bunch of red blood cells and white blood cells. So those have to be produced constantly, millions every day, in order to supply your blood with the cells that it needs to do its job. One other kind of strange one. In the middle, I've drawn this little dotted bunch of stuff. And that's other marrow, that's yellow marrow. That actually stores a bunch of fat. Fat that we usually don't use. It's kind of emergency reserves. When you have enough, then your blood will transport some of that into the middle of bones, and we only withdraw it if we really need to. Usually we use the fat under our skin, but we will also use the fat inside of our bones if we really, really need energy. And that wraps it up for the skeletal system.